Hi everyone, this little video covers what you need to know about the replication of DNA for higher human biology. It also covers the same stuff that's in higher biology as well. Now what you need to know for this section of the course is you need to be able to explain the process of DNA replication, understand the function of DNA polymerase, primers and ligase in the process, and also understand and describe the terms leading strand and lagging strand. Before we actually look at the process of DNA replication, we have to consider why it's important. Why do we even care? Well, as you will have heard 1,000 times, DNA is the blueprint of life. It allows the cells to carry out all the cell functions that they need to do. And when a cell divides to produce more cells, that DNA must be passed on. So DNA replication occurs before cell division. If there's any errors in DNA replication, the daughter cells that are produced will not be able to function properly, and this could cause massive problems for the organism. Now we're just going to go over the process of DNA replication. Now, I'm assuming we're all up to speed on the structure of DNA, and we've got that here. This diagram here shows us the structure. It's a double-stranded helix, okay? And what's important that you'll all be aware of is the base pairs being joined together by their hydrogen bonds. So stage 1 of DNA replication is we have to unwind the DNA, and to do this, the hydrogen bonds are broken between our base pairs. And to break those bonds, ATP is required. Okay. Once the DNA is unzipped and we've got our bases exposed, replication will occur on both sides. So on strand 1 and strand 2, they're going to replicate. Okay. So the DNA has been unwound and two strands are exposed, exposing the bases. We're going to look at just copying one strand. So if you've got our two strands of DNA and they've unwound and they're open, we're just going to look at copying the one strand, okay? The same thing is happening on the other side, but in a slightly different way, and we'll look into that later. So to copy our strands, we need something called a primer. And if we look here, this primer is just this little green section, and it's basically a small strand of nucleotides together, and they almost target an area of the DNA to start replicating. And the primer is really important. The primer must come in before replication occurs. Once the primer is there, an enzyme called DNA polymerase here will come and find the primer and basically just start adding on the complementary nucleotides to the strand of DNA that's exposed. Okay, so there'll be lots of A's and T's and G's and C's, adenine, thymine, guanine, cytosine floating around, and our DNA polymerase is going to add those on to the new growing strand. What is important is DNA polymerase can only add nucleotides to the 3' end of the strand. So if we look here, oh, where's my pen? We've got a 3' end there, but our new strand, we've got the 5' end. So DNA polymerase is adding the bases, or the nucleotides, onto the 3' end of the new strand, the growing strand. Once we've got all our bases lined up, we now need to form hydrogen bonds between the base pairs. So we've got the old strand, we've got the new strand, and we're going to have to get our hydrogen bonds in between the two strands. This is going to bring our two strands together. Okay. Once we've got our hydrogen bonds in there, what we'll then have to do in the new strand is those strong covalent chemical bonds will have to form between the nucleotides. Remember how DNA has got hydrogen bonds between the bases, but between the deoxyribose sugar and the phosphate of the other nucleotide, there's a nice strong covalent bond. So in that new strand, those strong chemical bonds are going to have to form as well. Now, something you'll remember about DNA is it's got anti-parallel strands. So one strand of DNA runs from maybe 5' to 3', and then the other strand will go from 3' to 5'. Now, if DNA polymerase only adds on to the 3' end of the growing strand, it means one of our new strands of DNA is going to be made in chunks. Okay? More on that later. Just believe me, it's made in chunks. And these chunks of DNA, if we look down here, we've got one chunk of DNA there, one chunk of DNA there. These chunks need to be glued together, and it's ligase, a special enzyme that glues the chunks together. Okay, so on the lagging strand, again, more details to come soon. Ligase joins fragments of DNA together to form a complete strand. Whereas on the leading strand, DNA polymerase can add on nice and smoothly, the nucleotides on nice and smoothly, so there's no need for ligase because it's all just one continuous strip. Now, this leads us to our final thing about replication of DNA, the actual process here, is that we say that DNA is semi-conservative, because if you've got your one original strand of DNA, got double strands here, and then it separates, this is from the original strand of DNA, and this is from the original strand of DNA. It's conserved, it's passed on to the new strands, because this side will make our new strand here, and now we've got a double-stranded molecule where one bit is from the old bit of DNA, and this bit is new, and whereas on this side, we've got again, from the old bit of DNA here, and then the new bit forms there. So it's semi-conservative because half of it is conserved when it's passed on. Now we're going to look at the direction of replication, because I mentioned there in the last slide that the two strands of DNA, when they open up, they replicate a little bit differently. We're first going to look at what we call the leading strand. This is almost the simpler one. So if DNA polymerase adds nucleotides to the 3' end of the new strand, this means that DNA polymerase can continuously add nucleotides as the DNA is exposed further, as the bases are exposed further. So basically, if you imagine this, right, here's my DNA, it's unwound, and now we're breaking the hydrogen bonds. So it's opening up like this, and that's our replication fork, remember, the V here is the replication fork. The bases here are exposed, okay? But as we open and break the hydrogen bonds, as we open up our DNA more, we're exposing more bases. And on the leading strand, it just so happens that the new bases that are being exposed are the old DNA's 5' end, meaning it's the new strand's 3' end. So on the leading strand, as the DNA opens up and unwinds, DNA polymerase is just adding on the nucleotides as they open up more. So it's just continuous, okay? That's our leading strand. It's going to happen a little bit faster than the other strand that happens in chunks. Hence the name is called leading. It's leading the way. So now that takes us to the lagging strand, the other strand of DNA. This is the one where it's the 5' end of the new DNA strand that's exposed when the fork opens. So as the hydrogen bonds on the original DNA break and expose our strands, the lagging strand is the one that's replicating the 3' to 5' end of the DNA. So if you look at this diagram, we've got the original one is 3' here and then 5' here, and our lagging strand is replicating the one that runs 3' to 5', okay? This means that the new DNA strand, the growing DNA strand, is actually running from 3 to 5'. DNA polymerase can only add on the 3' end, so it can add on continuously. As our fork opens up, it's got to wait a wee while to have enough bases exposed for DNA polymerase to jump in. For example, if you look at this diagram here, we've got DNA polymerase starting there and then working down. And then as more bases are exposed, it moves back up to here and it works down because it's got to add on to the 3' end. So our lagging strand is getting replicated in chunks. And as I said earlier, these chunks or fragments of DNA then need to be joined together with ligase. Okay, so all along the lagging strand will just be replicating chunks. Ligase will then come together and join those chunks up. Now the next few slides are normally what I might get the pupils to note down in class, but you might already have done that just now, or you maybe want to pause it and do it, or wait till the end. It's up to you. Right, to summarise, what do we actually need for DNA replication? Right, you obviously need DNA. We need a template. You need something to copy from. Press template. You need free DNA nucleotides. You need those nucleotides for the DNA polymerase to actually grab and add on to the new growing strand. If we didn't have any free DNA nucleotides, then we couldn't make more DNA. Okay. Now the two enzymes that were required were DNA polymerase, which adds the complementary nucleotides to the 3' end of the growing strand, and also ligase, which is the glue, the one that glues the lagging strand fragments together. We also require some ATP, because that's required right at the start to break the bonds of the DNA, break the bonds between the bases. And of course you need our primers, which are the short complementary sequences of nucleotides that allow the DNA polymerase to actually start the replication process. To summarise DNA replication, first, DNA is unwound. Remember, it's a helix, it's unwound. Hydrogen bonds between the bases are broken and you get 2 template strands. Primers bind to complementary sections of DNA, and this is required for the DNA polymerase to actually start, the enzyme that comes in and adds free nucleotides to the growing strand. Remember, only on the 3' end. This means that on one strand it's a nice continuous addition of nucleotides, that's our leading strand, but on the other strand, it's going to be our lagging strand, it gets done in fragments. These fragments are joined together by ligase. That's the end of my little summary. Hopefully I've explained the process of DNA replication and given you the function of DNA polymerase, primase and ligase, and you now understand the differences between the leading strand and the lagging strand a little bit better.